Hi my dear students, welcome to my biology class. Today we are going to learn the next experiment which was conducted by Alfred Hershey and Martha Chase to prove the genetic material is DNA. They conducted experiments with the help of bacteriophages and E. coli bacterium. What are bacteriophages? The viruses that infect the bacteria are known as bacteriophages. There were 3 steps in their experiment. They were infection, blending and centrifugation. They used the 2 types of radioactive labeled materials, the chemicals like sulfur and phosphorus, to understand how the bacteriophage infects the bacteria, what happens during infection, whether the protein or the DNA get into the E. coli bacterium or bacteria while infection. First of all, they grew the bacteriophages in 2 different medium. A group of bacteriophages in the radioactive sulfur medium and other colony grew in the radioactive phosphorus medium. And what happened? What was the result? The bacteriophage were lived with the radioactive sulfur medium. The protein coat of this bacteriophage became radioactive. But in the case of this bacteriophage, instead of protein coat, here the DNA became radioactive. Do you know why? Sulfur is the component of the protein, so that sulfur could incorporate with the protein components of the bacteriophage, this protein capsid of the bacteriophage. Here, phosphorus molecule is the component of the nucleic acid, so that it could incorporate with the DNA of this bacteriophage. So here the difference is, here the protein coat became radioactive, here the DNA became radioactive. Such 2 different types of bacteriophages were allowed to infect the E. coli bacterium separately. Let us examine the first case. What is the result of the first experiment? During the infection, they allowed this bacteriophage, the protein coat, the radioactive labeled, radio-labeled protein coat with this bacteriophage infected the E. coli. After infection, this mixture were agitated in blender. That procedure is called blending, mixing. So, at this time, what happened? The genetic material of the bacteriophage could get into the E. coli bacterium, so that the capsid of the bacteriophage could remove or separate from the E. coli bacteria. Finally, these were centrifugated. Then they got a result. They got 2 fractions. One is supernatant and the other one is the E. coli bacterium. They could detect the radioactive labeled sulfur in the supernatant. No radioactivity detected inside the E. coli cell, E. coli bacterium. What does it mean? During the infection, what happened here? The DNA, the genetic material, could get into the E. coli bacterium. It was not radioactive. Here the radioactivity was detected in the protein coat of the bacteriophage. So it concludes that protein is not the genetic material, DNA is the material, genetic material, which get into the E. coli bacterium while bacteriophage infection. In the next case, they got the same conclusion. Listen me. They allowed this radioactive phosphorus labeled bacteriophage to the E. coli bacterium. After infection, blending and centrifugation, they got a result. At the end of the experiment, they could observe the radioactive DNA in the E. coli bacterium. That means the E. coli, in the E. coli there were many number of bacteriophages. That means the DNA entered into the E. coli bacterium, so that no radioactivity in the supernatant, and but they could observe the radioactivity in the E. coli's body. Got it? The DNA incorporated with the DNA of the E. coli, and it started to produce the progenies of the bacteriophage. So it could cause infection in the bacteria. So this is the way they proved that the genetic material is DNA, not the protein. This was the confirmatory experiments which showed that DNA is the genetic material. DNA is the genetic material which transfer from bacteriophage to bacteria during infection. Why the DNA is considered as the genetic material? This master copy can store the information in the form of nucleotide base pairs. It can store all the information with regarding the complete features of an individual organism. So it is the hereditary material or the genetic material. Dear students, I hope you have understood the experiment well. With this, I conclude my session. Thank you and have nice moments.